over 150 people online now between the panelists and the attendees, and the number keeps going up. So welcome back everyone to the public session of the EMSL Summer School. And we have a couple more great presentations this afternoon before we finish up for the day. I think especially for those of us who have been in all of the sessions today, our heads are spinning a little bit with lots of information. But it's been super exciting, and it's been fun seeing some of the pictures coming in that Andrea's been posting on our EMSL Twitter site. In fact, I wanted to share one that I thought was fun. We had one from Rebecca Prescott, who told us that she was traveling from Scotland to her home in Hawaii when she got stuck in Florida on a horse farm. So she sent us a picture of herself hanging out with a horse and participating in the summer school. So check out the Twitterverse and send us your pictures to Andrea if you want to share with us. So with that, I'm going to turn it over to Kelly and have her introduce the afternoon speakers.
Great, and I'll keep this short so we have max time, but I'm super excited about you guys being able to hear from both of these speakers. Our first speaker is Rekha Seshadri, and she is representing the JGI. Rekha does a lot of the stuff that we are actually doing this afternoon in terms of functional annotation and other things, and has a pretty impressive Google Scholar page herself. But what we've asked her to do is come here and speak on behalf of the JGI. And so we alluded to the role that JGI is playing in this Wonders program in terms of these community sequencing projects. But we also know a lot of you are early career, and so we thought you could really benefit from learning more about the DOE JGI capabilities directly from an expert. And I can tell you myself, and it's others on this panel who are also earlier in career can also tell you that they've really benefited from both JGI user support as well as EMSL support. So we really wanted to kind of end the day letting you know what kind of opportunities there are and for someone who doesn't have to be a metagenome person. The other speaker is Dr. Eoin Brodie from Lawrence Berkeley National Lab. And Eoin Brodie is going to really teach us how we can take like his tools and how we can take kind of the annotations and what these genes are or aren't and how to scale those into a much more quantitative framework. And I myself am super excited to hear this talk. So both kind of using omics in many of the tools that we use today, and you'll probably hear a lot of the same vocabulary, but giving you different kinds of information. And so with that, I'd like to welcome our first speaker. Rekha, it looks like your slides are up and you're ready to go. So do you want to just take over?
Great. Thanks, Kelly. Okay, the Joint Genome Institute, or JGI. Everybody hear me okay?
Yes, you sound great.
Okay, thanks. The Joint Genome Institute is similar to EMSL. It's a Department of Energy user facility. And basically, user facilities are funded by the Department of Energy Office of Science to enable access to large-scale science and advanced technologies and provide access to a worldwide community of users. So the JGI itself was established during the time of the Human Genome Project. They were actually responsible for 3 of the chromosomes as part of the Human Genome effort, and they were established in 1997 and currently located on the Lawrence Berkeley National Lab in Berkeley, California, and we have 280 employees. So our mission is to serve as a genomics focused user facility supporting DOE mission areas, which is bioenergy, carbon cycling, and biogeochemistry. So I had some back and forth with Kelly, and I've sort of contracted and expanded this presentation. So this is where I settled. I'm going to provide some information on JGI's user capabilities, so our portfolio of products. And I have some within parentheses because I'm not going to go over all of them. I just cherry picked some that I thought would be, most synergistic with the topics that you guys are learning about in the summer school. But there are definitely other capabilities, and I have links provided so you can check those out at your leisure. And then the second part is just talking about the user programs. And these are basically program calls where you can send a white paper and propose to use those capabilities. So you get free access to all the capabilities, the sequence data, and some of the computational analysis if you're approved, if you're an approved user. And then the third part of the talk is a user interface platform that we have for comparative genomics and comparative metagenomics called Integrated Microbial Genome. So I'll touch upon that briefly. And if I have time, if I'm not going over, I might do this primer on annotation since we've talked a lot about, KEGG and DRAM and, all the different pipelines that are used to do functional annotation. So I'm going to attempt to get there, but if not, these slides will be made available to everybody. So you'll be able to sort of hopefully learn just from looking at the slides later on. Okay, so capabilities, some of them. So as I said, we're here to serve sort of these DOE mandated mission areas, and serving those areas, we're divided into 5 different programs. We have the microbial and the metagenomic program. We also have a plant program, a fungal program, and probably the most recent addition in the last 5+ years is our DNA synthesis program. And really what I want you to take home at the end of the day is that we are definitely known for our DNA sequencing and our advanced DNA technologies, but we do have a lot of capabilities beyond sequencing. And part of the mission, the way I like to think about it, is that part of the directive really is to constantly evaluate and benchmark emerging technologies, which in our case has been sort of sequencing platforms, the software that goes along with it for the computational analysis, looking at other types of emerging technologies, and then passing on the access to those technologies and to the lessons learned and the know-how to our worldwide community of users. So sequencing is our bread and butter, and I would say that we have 2 primary platforms. This is not to say that we haven't evaluated or do not have other sequencing platforms, but these are our 2 primary platforms, which is the Illumina and the PacBio. A significant amount of data is generated from primarily non-human or environmental samples that arise from as I said, non-human environments. And each of these platforms is really applied to the products that are listed below each one. So I think there was a question earlier on today about long read technology and something about coverage. And I just kind of briefly said that, so PacBio in particular, we use for generating de novo genomes of both isolates, of both prokaryotic as well as fungal and plant genomes and such. And sometimes it's used in combination with Illumina-based sequencing. But we do have, we use the Illumina primarily for resequencing, generation of metagenomes, metatranscriptomes, and our single-cell sequencing. And the PacBio, on the other hand, is used for some of the products that are shown here. But as I said, we have many products that go beyond sort of these sequencing or these advanced sequencing products, like the single cell. We also have MethylSeq, DAPSeq. I don't want to go on about different acronyms that you may or may not heard of, but we have a lot of capabilities. And please visit our product offerings page to see what all of those products are. But what I want to leave you with is that we have also capabilities beyond sequencing, which is our DNA synthesis program, for example, which allows the user to synthesize genes or pathways. If you're interested in functional characterization of genes and pathways, is very popular with biosynthetic gene clusters for secondary metabolites, for example. We also can synthesize combinatorial pathway libraries for metabolic engineering purposes. And we have individual program calls that allow you to use some of these capabilities. We also have metabolomics, and I thought that I would touch on that briefly because I think it might be of interest to this audience. Okay, so moving forward. I have some beeps happening. I hope my volume is still good. But so I just cherry picked one or two things to sort of focus on. And one of them is our brand new project, which is stable isotope probing metagenomics. And really the idea here is allowing you, the user, to link microbial identity to metabolic activity based on the uptake of isotope labeled substrates. So what the schematic is showing is that you would have samples from the environment that are probably incubated or enriched under certain conditions, and you provide then the isotope labeled substrates for its uptake, and the actively dividing members in that enrichment would then preferentially incorporate that label into its nucleic acid. And at this point, you would extract the DNA basically and send it to the JGI. And we would take care of the partitioning of the fractions and doing the quality control, and then it would run through our metagenomic sequencing pipeline. And as I said, this is a brand new project, and we're anticipating that we'll be able to accommodate 4 projects per year. Since it is brand new, I don't know if you can find any information through any of our program calls, but if you're interested in taking advantage of this opportunity and you want to learn more about the details or how to put together sort of your study design, please contact the 2 people that are responsible for bringing this particular pipeline to fruition, and that's Rex Malmstrom and Emily Elo-Fedrosh, and that their emails are provided, and they'd be very happy to talk to folks if they're interested in taking advantage of this particular capability. As I mentioned, we have metabolomics, and this is all under Trent Northen's able hands, and through the metabolomics program, you gain access to LC-MS, MS-based metabolite profiling in both targeted and untargeted analyses. And again, I've provided a link. So please check this out at your leisure. I'll talk about the user programs. In particular, we have a user program that allows you to use JGI's capabilities complementarily with EMSL's user capabilities, and it's called the FICUS call, and we'll talk about that in a second. What it allows you to do is sort of submit one application to both user facilities and take advantage of the individual capabilities in both location under one study. Okay, so you can gain access, as I said, to all these products, some of the ones that I talked about, and an much more expanded set that I didn't talk about, by submitting a letter of intent. Generally, that letter of intent is really to check to see that the proposal that you submitted is of DOE mission relevance. I mean, it's really a check to see that it falls. It's not a study about human genomes or something like that, but it's of DOE mission relevance. And at that point, you're invited to submit a white paper proposal. And then those white paper proposals then undergo a peer review process by the scientific user community. And so, again, I'm listing a subset of these particular user programs. One of them is called the Large Scale Community Science Program. And really the emphasis here is on large scale projects. We have a cap of 3 terabases for Illumina sequencing and 100 GB for PacBio sequencing under the auspices of this particular program per project. And all the capabilities are offered. So the things that I mentioned, the DNA synthesis, the metabolomics, some of the advanced epigenomics, all of those capabilities are offered through the large-scale community science program. And as I said, you have those really large caps on data that you can reach for. And these are some of the specific theme areas that we've been encouraging proposals to sort of focus on. So I'm not going to read those out. I'll just point you to this, and this sort of gives you an idea of sort of the success rate of individual. And so the success rate for the large scale community science program is 35%. So in terms of the proposals that are ultimately approved, it's pretty good at 35%. The other program call that I wanted to highlight was the new investigator call. And here the emphasis is really to favor first-time investigators that have never had a funded proposal through the JGI before. So it really targets sort of early career scientists. And I guess the objective at the end of the day is sort of preliminary data generation. So it allows you to sort of develop a pilot project, and then you can use that information to then submit for a large-scale CSP. However, unlike the large-scale CSP, not all of the products are available under the new investigator call. It's limited to the sort of subset that I've shown before. So it still allows you to do the metagenomics, the metatranscriptomics, both for isolates, for microbiomes, and also the DNA synthesis and metabolomics product line is offered through the new investigator call. And then, as I already mentioned, we have a joint call called the JGI EMSL FICUS call. And please check out our website and probably EMSL's website for sort of timelines of when proposals are being accepted and when due dates are. But this particular call gives you access to capabilities at both facilities with just one application. And so I've listed, for example, for EMSL, we have high throughput proteomics, advanced imaging, and complex sample preparation. And at the JGI, it would be sequencing, DNA synthesis, and metabolomics with LC-MS based methodology. Okay, so one more about JGI. I've given you a link to sort of our user programs. You can explore all the different types of program calls that are out there. And I also wanted to mention that any investigator is eligible regardless of their institution or national origin. So we do have a worldwide community of users. All that we need is for it to be of DOE mission relevance. And if you have any questions about any of these user programs, please contact Tanya Woike. I have her email provided here below. She's serving as our interim deputy director for user programs, and she'll be very happy to chat with you about any questions that you might have about submitting a proposal to one of our user programs. Okay, so under the auspices of all these user programs, JGI has generated a lot of data. And what you're seeing here is JGI's contributions to public databases. And you'll see that, a significant piece of the pie, both in terms of plant genomes and other types of microbial genomes are contributed by the JGI. All of these data can be accessed to dedicated portals. So for example, for plant genomes, we have something called Phytosome, and we have Microcosm for fungal genomes. And it's not just a repository for the sequence data, but it's also got tools for comparative analysis. On the prokaryotic side, for isolate genomes and metagenomes, we have something called IMG, Integrated Microbial Genomes and Microbiomes. And I'm just going to show you some of the some of the, not the tools. I mean, it's a large behemoth, so it's kind of hard to cover it all. But I just want to make you aware that this tool exists, and it's a data management system for comparative analysis of sequence data. So basically, all of this JGI generated data does end up in IMG. So we have a dedicated assembly team, because we are a sort of high throughput, high production type environment. We have hundreds and thousands of samples that need to be assembled and annotated. And all of those data get assembled and then annotated through the IMG annotation pipeline. So you'll see here, data sets that were generated by the JGI. For isolate genomes, we do go to GenBank and periodically update public isolate genomes from GenBank or from RefSeq. In terms of metagenomes, we have, again, primarily JGI generated data, but we also have public metagenomes, things from the Human Microbiome Project, for example, and some other genomes. It's not everything that's in GenBank, but there's still about 23,000 metagenomic samples. And we also have these other uncultivated entities, if you will, like the single particle sorts, cell enrichments, and metatranscriptomes. Okay, so there's a lot of data that you can look for, explore, and there are tools for comparative genomics. We conducted a webinar series relatively recently, and so all of those videos are available if you're interested in checking out what the possibilities are. Again, something that I wanted to highlight, because we're talking about metagenome bins this morning, is for all publicly available samples, all publicly available metagenomic samples, the initiative is to provide sort of pre-computed metagenomic bins. And this is the schematic that shows you that we run MetaBat to generate these bins. We do a certain amount of QC to remove sort of offending contigs that don't fit in that pre-computed metagenome bin. And we perform a CheckM based evaluation, just like you did. And only the high quality and the medium quality metagenome bins are then loaded into IMG. And so at present, we have over 101,000 bins that have been predicted from over 12,000 metagenomic samples. And all of these bins can be retrieved or explored based on their taxonomic affiliation, based on both an IMG's centric methodology, as well as your genome taxonomy database that you used probably this morning, GTDB-TK taxonomy, or by their underlying environmental or ecosystem metadata. And then you can do sort of these cross metadata searches using a bin search tool. So here's just a screenshot of what it looks like. So I'm showing a screenshot where I've sort of navigated down to 152 metagenome scaffold bins assigned to the genus Nitrosopumilus under the Thaumarchiota. And you can click on this number and it'll retrieve a list of all those bins, along with a lot of information about the content of those bins and the CheckM statistics. You can also peruse the bins by their ecosystem classification. So in this particular example, I've drilled down from terrestrial samples down to permafrost, and it's telling me that there are 768 bins that have been called from samples that are attributed to permafrost. And as I mentioned, we have a webinar series. I have links to all of these in the PDF document that I think you will be, either you already have or will receive shortly. So check out the webinars, and maybe it'll be of some value. Okay, I don't know how I'm doing on time, guys. I'm going to assume that I'm still good, and I'm going to soldier forward unless somebody tells me to stop.
Just about 9, well, yeah, it's probably 6 or 7 minutes if you want to leave a couple for questions.
Okay. All right, so other than JGI sequence data, we also encourage people to submit their own metagenomic data. So whether it's sequenced by the JGI or not, we do accept submissions within IMG of external data. So you as an external submitter might have assembled something in KBase, for example. You can submit those assemblies to the IMG annotation pipeline. Here's some details about what you would need in order to do that. And here's just a schematic of the IMG annotation pipeline. I don't want to delve into these details. What it gives you on each side is this is the structural annotation pipeline. So it's the gene calling. I think in the morning, people mentioned Prodigal. This is the structural annotation pipeline where the open reading frames are called. We use two different gene calling methodologies. There's a standard operating protocol also that's available. And then this is the annotation pipeline. So it gives you an idea of the tools and the databases that are used to do the structural and the functional annotation. And then all of that content then gets submitted to the UI system, and it's then sort of presented there in sort of a comparative context and in the context of various tools where you can do some comparative analysis. So here's, I guess, the primer part. I just wanted to briefly talk about structural and functional annotation. Just to familiarize everybody, although you've learned this before, so I'm just going to go over this quickly. So the purpose of structural annotation is to get at protein coding, at coding sequences, not just protein coding. It can be regulatory or RNA sequences, tRNAs. So structural annotation really is just looking for the features of a gene. So looking for canonical starts and stops, and looking for signatures or ribosomal binding sites, and calling these open reading frames in all six directions, right? In all six frames. And the ultimate purpose of structural annotation is to find the coding sequence. So structural annotation is not open reading frame finding, but the purpose is to find coding sequences. And in prokaryotic genomes, this is relatively straightforward. We try to go with the longest open reading frame that's usually correct. And we try to resolve any overlaps. In certain high GC genomes, this can be an issue, but that's the purpose of structural annotation. But when people think about annotation, they like to think about this part, which is functional annotation, where you're getting from sequence to an idea of the enzymatic function, an idea of the underlying biology. And so to do this, you search various types of databases and you look for similarity. And based on that similarity, you transitively assign a function. And so there are many, many archives of databases, and you've heard about a lot of them today. So I'm going to skip this part. It's just talking about how you have bioinformatics curators around the world sitting around and trying to record information from publications in a structured way that can be represented in a database and that we can then use to do our comparisons. Searching these databases, there are two general methods. One is everybody's favorite BLAST, which is called the pairwise method. And all that is you have two strings. In this case, it's amino acid string sequences, and you map these two strings, allowing for certain substitutions, a certain amount of insertions and deletions, and then you use kind of this best hit approach to parse the information, and then you transitively assign function based on that, and BLAST is an example of that. But there are many other tools that are being developed. As the size of databases grow, there's always a trade-off between sensitivity and speed. And so some of these other tools that you may or may not have heard of try to solve that problem. And databases that you do pairwise searches against are obviously NCBI-NR. I'm sure everybody's been there at one point to run a BLAST job against their NR database. So within IMG, we also have an IMG-NR database that we do a pairwise search against. And as Dram already mentioned, We also do a search against the KEGG orthology database. So here's a pathway map for that. The second method for looking for similarity is this HMM search that you heard about this morning. These are profile-based methods. And the difference between these is that first, the curator develops a really good alignment. So you take a bunch of reference sequences that are well characterized, well known, generate a really good alignment, and then compute the statistical probability for the amino acid in those positions in the seed consensus. And so here's a schematic again. You have a multiple sequence alignment, and then you have the consensus, and you compute the statistical probability, and you build a profile such as a hidden Markov model or a position-specific scoring model. And hopefully everyone can appreciate now that this is computationally a lot cheaper than having to do a blast search against an entire IMGNR database or an NCBNR database. Instead, all you're searching is these consensus models. And it also can be more sensitive than a pairwise alignment. So very frequently, you might not get a pairwise hit against an NR database, but you might get an HMM search result. And so again, there are many different databases that are established and updated that are out there. And here are some examples in addition to the ones you heard about in the context of RAM. So within IMG, we perform HMM searches against NCBI's COGS database, the TigerFam database, the PFAM database, and then more recently, we've added Cat FunFam, SMART, and Superfamily databases. Okay. You might ask, why do we have so many different databases? And maybe somebody's already answered this question before. They're not all perfectly overlapping. They have individual strengths and individual limitations, if you will. And so there is a reason why we try to maintain annotations, all of these different types of databases. And the reason for that is sort of maximize the opportunity to make an inference.
It's 4:23.
Okay, thank you. I'll wrap up. So all the data can get submitted to the IMG annotation pipeline, and on the UI side, you have access to various tools that allow you to sort of explore your data and do some comparative analysis. I will leave with this slide. Just to let you know, we have an annual user meeting. Obviously, we didn't have one this year, but I think we should be back. Well, we're all fingers crossed we will be back on track next year. And that's an opportunity to hear about exciting new projects from our users and from the scientific community at large that use sequencing-based approaches. IMG has its own hands-on on-site workshop that'll be here at Lawrence Berkeley National Lab. And we have a webinar series that you're welcome to check out. There's a YouTube channel that has, I think, eight videos posted there. Okay, I'm going to stop. I apologize if I went at a breakneck speed. I'm assuming there'll be questions. I don't know if we have time to take them now, or I can try to answer them via chat.
So yeah, we can definitely, you can look over the chat, but one of the key questions we can answer real quickly was, well, a couple were related to access for users. And so one you already answered very well in your presentation. One user asked before your slide about international users and if it was open to non-US scientists. And so clearly the answer to that is yes. Another one asked about supporting projects funded by DOE agencies other than BER. So for example, ERE, the renewable energy or fossil energy offices, and if you could maybe give a little insight on that.
I think the answer is yes. Perhaps I'm not the best person to answer that question. Tim, I don't know if you know more than I do.
Well, I think, and Nancy answered in the chat too. I think it's really an issue of, like you said, for the international or anybody else, as long as it's aligned with BER and DOE science missions. That's really the issue, right? So, obviously if it's not aligned with those missions, the answer is no, but if it is, it really doesn't matter so much who's funding the actual project as much as it is about the alignment. And I think that's also true for EMSL as well. So, and obviously, thanks for, pointing out the joint EMSL JGI call. That's, I think, been a really successful avenue for users to be able to access both facilities with one proposal. So great. So thanks, Rekha, and do appreciate the presentation. We'll be posting these again as soon as we can. And if you have questions, more questions for Rekha, please go ahead and post those in Discord, and she'll be looking over there and chatting with you there. So we're going to move on. We have a little bit of time left for one last presentation. And so we have Eoin Brodie, Ulas Karaoz, and Gianna Marschmann presenting on trait-based modeling. So take it away, Eoin.
You just come off mute. Great. Well, thanks very much. Can you see my slides?
Yes, it looks great.
Yes. Great. Okay. Well, first of all, I want to say thank you to Tim and Nancy and everybody else and Kelly and everybody else for organizing this. I think this has been a phenomenal session, and the participation so late in the day from all across the world is really great to see. We're going to jump in sort of downstream of what you've heard earlier on today, and we're going to make the big assumption that you have quality genomes from your system, so that whether that's from single cell genomes, from isolates, or from curated metagenome assembled genomes. And by now, you know the amount of work that goes into that. What we're trying to do here is to understand microbial communities assembly, the implications for biogeochemical processes, and we're trying to do this in a scalable way, one that both honors the complexity of biology, but also allows us to test ecological theories across different scales. And I think this presentation, I think, is emblematic of how this is a multidisciplinary challenge. I'm a microbiologist and microbial ecologist. Ulas Karaoz is a computational biologist, and Gianna Marschmann is a biogeophysicist, and each of those labels are interdisciplinary. And so together it takes us and then an army of other people as well to make this work possible. I want to begin with a really great review in Trends in Microbiology from Geneviève Lajoie and Steve Kemble, and trying to point us in the right direction of how to make the most for trait-based approaches in microbial ecology. And one of the first parts of this is to define what we mean by a trait. And I've just borrowed some text from their publication here, and this really takes the perspective of a trait from the concept of fitness. So really a trait is anything that can explain differences in fitness among individuals. Okay, so if we can agree on that, we can move forward. Now, traits can be simple or complex. It could be a single gene, or it could be a property of a whole organism. And we'll get into that difference in trait complexity as we go through. Another goal, to be able to scale and interpret traits in the context of microbial fitness, is to be able to reduce the dimensionality in the large diversity of traits. Now, microbes have adapted and optimized many different ways to overcome the constraints of mass and energy balance in optimizing how and when and where they perform certain activities, or the balance of certain activities. So that leads to a large amount of trait complexity. And our goal is to turn that complexity into adequate simplicity, and adequate depends on your scale. It also depends on your question. What we'd also want to be able to do is to take this information, these complex traits in genomes and across many organisms, and to derive a mechanistic explanation for ecological patterns that we observe, identify if there are generalizable combinations of these traits and their trade-offs. I'm not going to go into what those trade-offs are. There's an asterisk at the bottom there. But how they appear across scales. And also, if we do a good job of this, when we recombine this information into models, can we recreate and predict emergent processes? So most of the things we observe in terms of biomass growth, CO2 respiration, biogeochemical process rates, these are emergent processes. And can we recreate those through the interactions of traits and populations when we combine these in the local environmental or community context? So you can think about it like this. What we're doing here is we're inferring traits when we talk about genomes. In some cases, we have some phenotypic information to verify those inferred traits, and we have to represent the costs and benefits associated with those traits. Now, the fitness of an organism, the traits are a feature of an organism, but the fitness is not just a feature of an organism, it's the feature of the organism, its neighbors, and its environment. And together that controls fitness, that controls the assembly of communities, which then feeds back into the process rates, that changes the environment, that changes the fitness. And so we repeat this cycle. So we need an environment, a computational environment that represents this series of events. So when we talk about traits, what sort of traits are we talking about? What can we infer from genomes? Well, we can start off with what we've heard about today already. These are our metabolic traits. So the inner workings of a cell related to acquiring resources, your transporters, enzymes, siderophores, etc. How a cell generates energy through respiration, fermentation, what electron donors, acceptors does it use? How does it deal with stress, and how does it invest in stress tolerance? And how does it deal with antagonism, whether it's the protagonist or the antagonist? How does it defend itself? So these are the traits we commonly think of. But we also have more complex traits. So the size or the volume of a cell determines its surface area to volume ratio, and its effectiveness of obtaining substrates from the environment. How well do substrates diffuse through its membrane? Its cell wall composition determines its rigidity, its chemical demand, and its cell carbon demand is also a function of its growth rate and its cell volume and size. And then moving up in complexity, we have life history traits, and these are traits that are a product of evolutionary history of the organism, and they're imprinted in the genome. For example, maximum specific growth rate, you can predict the upper bound on growth rate of an organism over evolutionary time by genomic properties. You can also predict the optimum growth temperature within certain margins of error from the organism based on how its genome has evolved over time. So we can use this information to predict what the lifestyle of an organism and its niche might be. Now, I like to use this analogy. We're inferring traits, and what we'd like to be able to do is then to take this trait inference and infer the ecology of organisms and how that either persists or changes across scales, and what are the rules associated with assembly across scales. And an example over on the left-hand side here, this is the NSF Neon Airborne Observatory platform. And this image here is a hyperspectral imaging of of multiple watersheds actually in Colorado. Dana Chadwick organized the ground campaign associated with this. And this image shows you that you have hotspots of plant nitrogen availability or acquisition across this watershed scale. Now, this is coming from spectral information on this aircraft, and this could be, in some cases, drone, in some cases, UAVs. You can zoom in closer, you can see a river here, and you can see, again, these hotspots of nitrogen acquisition in vegetation. And then when you start to provide context for this, also ground truthing, so you verify that what you're seeing via spectral information you're inferring is somewhat related to truth. When you overlay this with other information, such as geophysical information, ground-based measurements of water, chemistry, soil physical structure, etc., you can start to predict why different assemblages of plant species co-assemble and occur in these patterns across the landscape here. And that's ideally what we'd like to be able to do with microbial trait inference, be able to test theories across scales and then see do theories evaluated at this hill slope multi-meter scale, do they also appear at watershed or multi-watershed scales? So for microbial trait inference, we're kind of doing the same thing, except our lens is through the lens of a DNA sequencer. And we begin with complexity, and then we try and reduce this complexity as we move up through scales to explain patterns of microbial assembly and their contributions to ecosystem processes. So our genome prediction gives us the guts of the organism and maybe some biophysical traits. But then as those organisms start to interact in a three-dimensional matrix under the influence of other trophic levels like plant roots, in the context of changing hydrology in the landscape, we need to be able to consider how all of these factors contribute to the regulation of the fitness of the organism at this scale. And just like For vegetation ecology and field campaigns, this requires ground truthing. And in our case here, the ground truthing comes primarily in the case of genomes that we have isolated. Okay, so how do we go from genomes to inferred traits? I'm going to hand over to Ulas Karaoz to give us a primer on how we go about this.
Hi, I'm Ulas Karaoz from LBL. Hopefully everyone can hear me. So as Owen described, how do we inform a trait-based model with genomic data? That's our goal here. Essentially, what we want to do is to represent the microbial genome given a sequence with an inferred trait fingerprint. And as Owen went over, each trait here is represent one or more aspects of an ecological strategy, and we need to look into the literature to find either single or aggregate genomic signals in the genome that we can leverage. So starting from the bottom, the biophysical traits, especially the cell size, is a key trait that underlies many life history traits, and specifically how you use substrate efficiently. And we can get to that through genome size scaling. Next is the two key life history traits, which leave aggregate signals in the genome. The minimum generation time is known to be predictable from codon usage bias, and we can use approaches that look into amino acid frequencies and link them to optimum growth temperature. So next are several traits which underlie different dimensions of different strategies to be fit for environmental microbes. So a major one is under resource acquisition. So a microbe would invest heavily in in extracellular enzymes to break down complex substrates, complex matter, or it could invest in transporters for specific or a group of substrates. So these will, how much it invests in its genome to these classes of enzymes or transporters would represent the resource acquisition strategy. As you heard in very good detail and perfectly this morning, the energy generation is an important trait which determines what kind of energy generation pathways the microbe can use given the available substrates in the environment. And the next two classes of traits, the dimensions, ecological strategies, which leave genome signatures, are things that let microbes to be involved in antagonistic interactions or deploy defense strategies. And these typically manifest into genomes as investment in fatty acids, peroxides, and antibiotics, as well as CRISPR loci. And the stress response is known to be underlined by investment in osmolytes, chaperones, and extracellular polysaccharides. So once we can represent the genome in terms of these traits, we can use the parameters that you just saw. So we need to do this for hundreds for thousands of genomes. So we need a way to digest those genomes and transform them into what we call trait matrices in terms of these traits. So for this, we use a model-based approach where we start with IMG, the genomes and metagenomes in IMG. And as just described in the JGI lecture, we use this model-based approach to curate gene-level models. So these are not domain-level, these are gene-level models to detect loci that underlie these traits. So this gives us a curated database of HMM models, essentially. Then we take each of those models and then we determine trusted cutoffs, which tell us what kind of precision and recall we are expecting when we do the searches with these models. And we do that as much as possible, as much as these are represented in cake, by finding the cake by treating KEGG as the gold data set and by finding the homolog in KEGGs for these models, and then using those as a benchmark. And overall, about 80% of our models have both precision and recall about 90%. So just to, so so far I described how you detect the genetic loci, and then once you have these loci, then you have to turn those into some metric of trait. So either these can be either binary or investment in particular classes of things. So I try to exemplify this with transporters. So for transporters, we use the transporter classification database where you have transporter families and documented with their substrates from the literature. So we represent each transporter with an HMM model, and then they have that model as linked to substrate, which can be classified at different granularities. And we represent that inference, what I call inference here, from going from the gene content profiles to traits with a simple Boolean logic, which is reusable, reconfigurable from a user perspective. So here I give you the example of fructose transport, where there are three potential genes in transporter database, and with each subunit represented by a hidden Markov model. So you can reconfigure that rule as requiring all the subunits to be present, or just the substrate-specific subunit to be present. And when you take that the fructose transport to the upper level of monosaccharide transport, you can write a rule that will correspond to the monosaccharide transport, with essentially ORs all the different monosaccharides. So here's an example of what we can do with the trait matrices for the specific case of resource acquisition guilds. So what we want to do is with the trait matrix is to take that matrix for a set of genomes, where in here I exemplify it with isolated soil genomes, which we got from IMG, and then ask like how many guilds we can define given that matrix. So the answer is going to depend on how detailed you want to go and how much complexity your model can handle. So that's a parameter. So here I show you the trait matrix for these soil genomes and for resource acquisition traits, and then represent each genome as an enrichment score of that trait compared to all other genomes. If so, a red color means that that genome is is statistically significantly investing in these resource acquisition traits. So you see that blob on the top, these are all the genomes, which we call exudate user guild, which invest in transporters for exudate versus, so they don't invest heavily in extracellular enzymes for polymers. So we interpret this as a ecological strategy that manifests itself in this guild. So similarly, on the bottom, you have a guild for the complete opposite strategy, which is the polymer users. And you can see that taking these genomes from IMG, you have everything in between. So, so you can see that there are a lot of other guilds and like middle guilds that you can get from this. So you want to get a picture of how many guilds you need, like once you want to model this, and you can discover this with say k-means clustering to achieve a dimensionality reduction, where so in this case something like 26 guilds will cover 80% of the variation in this data set. But that's up to you really, like how you want to parse out this data given this tool. So once you have the guild, then we can, before going into the modeling, we can parameterize each of these guilds with the kinetic parameter through the minimum genome predicted minimum generation time. So you see that there's a difference in the minimum generation time given this data set and in the genome sizes, which become parameters that that's to be used by the trait-based model. And Owen is back to Owen. He's gonna describe more how we do that.
Great, thanks, Ulas. So the question is, once we've distilled these genomes down into traits at those different levels of complexity, what can we do with that knowledge? And the first thing here, I'm going to go back to this image. Ideally, what we'd like to be able to do is determine relationships between those traits, how those traits are linked within genomes and their distributions in space and time. And then that gives us some inference on what the niche of those organisms is. That tells us something about the factors influencing their assembly, but it also tells us something about the likely biogeochemical processes occurring in that physical dimension. The other thing that we're going to spend more time talking about here today is how to parameterize models. And by parameterizing models, then we can start to explore what traits of organisms are important when and where, and how trade-offs occur. And also then what are the process implications? So the implications for biogeochemical cycles, for example. And to do this, we want to share with you a vignette. So this is a study that combines a number of papers being published over the last few years and some ongoing work. And this is looking at the succession of soil bacteria in the rhizosphere. And I realize we didn't introduce all of the necessary terms to this point here. Here's the root zone of the plant. The area adjacent to the root zone under the influence of the root is called the rhizosphere. Areas out of the influence of the root zone directly over here would be termed bulk soil. And you might expect that organisms living in the bulk soil here would mostly subsist on dead plant material or decaying organic matter, soil organic matter, while those directly in the rhizosphere might subsist on root exudates, so the monomeric substrates that Ulas was talking about there for transporters. So a long time ago, Shengjing Shi did an experiment to follow different types of microbes and how they responded to a growing root versus the bulk soil environment. And Ulas's Nunes de Rocha developed an amazing resource of an isolate library that represented a lot of the diversity of the bacteria that were responding to this root growth here. Kate took this work a little bit further, and she matched up the patterns of OTU succession in the rhizosphere here with these isolates, and then did comparative genomics to see what were the features of the organisms that responded positively to the root and those organisms that were responding negatively to the root. And what she found was that the rhizosphere adapted organisms, against our expectation, seemed to have a slower maximum specific growth rate overall, but they had a higher number of monomer transporters in the genomes, particularly for amino acids and organic acids, and they had lower number of extracellular enzymes. So very similar to the guilds that Ulas described previously from the whole soil genome data set. Now, Kate, working with Trent Northern, explored the chemistry of the roots, and we saw that the roots were secreting different compounds over time, with sugars, organic acids being secreted during earlier root growth. And then she fed a cocktail of those chemicals to the plants or to the microbes, and identified which types of substrates were depleted from the media or used by the microbes. And Kate was able to show that, yes, substrate preference as predicted by genomes does appear to occur when we try and observe that. So the relative substrate preference of the organisms seems to be important to their fitness in the rhizosphere and is predictable from genome signatures. Now, Kate has taken this further and more recently has shown that, yes, the bulk soil bacteria have higher genomic allocation to extracellular enzymes, particularly for hemicellulose and oligosaccharide degrading. And interestingly as well, using proteomics has shown that bulk soil bacteria secrete roughly 10 to 20 times more enzymes than the rhizosphere bacteria. So that would be a cost to the bulk soil bacteria in this environment. Okay, so I'm going to hand it over to Gianna Marschmann now, and Gianna's going to tell us how we would represent these traits in a model.
Hi. I'm Gianna. I'm a postdoc in Owen's lab, and I'll use the remaining time in the talk to talk about how we represent microbial traits in a dynamic energy budget model, which is a modular model for metabolic organization developed by Bas Koyman and shown as a schematic here on this slide. It represents traits and trade-offs between traits in terms of mass and energy conservation as a main feature. So the way it works is that in the model, each microbe has a reserve pool made up of storage compounds, And these storage compounds are dynamically allocated on a stoichiometric and energetic basis to growth, maintenance, and other metabolic processes. Here, for example, by investing in extracellular enzymes or defense mechanisms against phages. So we can map genomic information that Ulas collected onto the dynamic energy budget model structure by finding appropriate genomic trait proxies for the model parameters that are highlighted in blue here. And because we have the genomes as a basis, it is then possible to assess trait performance given biophysical constraints, either on the scale of one single organism, for example, in the context of the size of an organism, and then assess its fitness in terms of biomass, growth rates, or carbon use efficiency, but then also to scale across different levels of organizations and test our genomic hypotheses for the relative fitness of emerging microbial guilds, for example, the positive and negative rhizosphere responders in an environment where the environment here, for example, would be represented by a network of different polymers, monomers, and mineral surfaces that overall interact with traits of organisms to influence biogeochemical reaction rates or pathways of soil organic matter formation. So on an organism level, in the dynamic energy budget model, cell size is a key physical constraint because it partitions the energy budget of a cell, as shown here on the right, into different fractions used for either growth, maintenance, or other metabolic costs that we want to include. So we infer cell size using the observation that it scales sublinearly with genome size across many diverse bacteria. And cell size is important because it scales the maintenance rate and the fraction of the total metabolism that is used for growth. So you're both highlighted in light blue. So also the maximum specific growth rate sets an upper bound of the growth rate that can be realized for an organism in the dynamic energy budget model. And we infer the maximum specific growth rate from codon usage bias as a genomic trait proxy. Also, the realized growth rate then depends on the capacity of or ability of the organism to synthesize structural biomass from storage compounds or the reserves inside the cell, which is itself a function of the maximum translational activity of ribosomes, which depends on RNA copy numbers in the genome. So cell size so not only describes the energy budget, but it also determines kinetically the microbial uptake of substrates of organisms in a diffusion limited environment. Here in the equilibrium chemistry formulation, the affinity constant K and the maximum specific uptake rate are the key fitness traits that determine the rate of of uptake for different compounds. And this rate of uptake depends on the density of transporter proteins that are distributed across the cell surface or the surface of a cell for a given size. Now, as we've seen by Ulas' classification, that transporters can either be general or specific. So we can define a network of uptake for different substrates per cell, and determine the importance or the investment strategy of an organism based on the gene frequencies of different transporters in the genome. So what we've done is what can we predict from biophysical constraint? So we use the allometric scaling relationships for how maintenance, growth efficiency, and protein synthesis rate depend on genome size and RNA copy number to predict the number of transporters on the cell surface that are necessary to grow at a given maximum specific growth rate. That scales with cell size, as well as a relationship between realized growth rate and cell size. So the isolates that Eoin described previously are plotted in green and magenta. so their size only differs across 1 to 2 orders of magnitude, and they generally fall into a larger size range. So what this means in terms of their growth strategy then is that because they're bigger, their larger maintenance costs can be offset by high growth efficiencies and high rates of biomass synthesis, mainly because larger organisms with larger genomes allocate an increasing fraction of their genes to biosynthesis. In terms of uptake, because larger cells have a number of transporters, we will see that the positive responders will have a higher maximum specific uptake capacity as well. So if we allocate the transporters based on the relative gene frequencies to the predicted number of transporters that have to be present to satisfy calm demand for cells, shows that rhizosphere organisms have a higher maximum uptake rate and lower affinity for organic acid uptake. But there's no significant difference, for example, in the preference for sugars, which is consistent with genomic traits associated with their soil niche from the vignette. But their fitness advantage is spatially constrained because the higher affinity constant requires them to stay near zones of high concentrations in the rhizosphere. So it might be important in the future to represent other traits like the ability to attach to a growing root or the ability to swim towards it. And we could again get this from presence-absence traits from genomes and by considering the energetics and scaling of cell movements with size.
Gianna, this is your 3-minute reminder.
Right, thanks. So this is my last slide because as it is, a difference in cell size and transporter diversity that lead to difference in substrate uptake only contribute a small amount to differences in relative abundance between positive and negative responders. So they explain only 4% in change in relative abundance. So moving forward, we'll have to include more traits in order to explain the relative fitness of gills, and can maybe once again look to the plant community where the explained variants of trait-based models saturate at 7 to 8 different traits that are represented in these types of models. However, I think what the potential is with the DB model is that we really have a formal way to explore genome-inferred traits either in isolation or in combination through model parameterization, which will then allow us to identify in this way the necessary trait complexity to explain the observations in a given system. I'll hand it back to Eoin for the final words.
Great, thanks, Gianna. And I just have a final wrap-up slide. I think one thing here, though, is that the signal seemed to say that substrate preference was everything. But when we explore that in a model scenario, looking at the differences in kinetics, the quantified differences in kinetics, it really only explains a small amount of variance, which means we need to add more complexity, more traits into the model to be able to identify what are the mechanisms leading to these successional differences. So just to wrap up, what we've tried to show here that we all know that genomes are a record of evolutionary history, but also of contemporary fitness traits. They're now considered the species unit for microbial ecology. They are our primary window into microbial function on Earth, but they are only hypotheses of function, and we must do better at testing those hypotheses using other methods, evaluating their phenotype. And we do need modeling approaches to be able to integrate and evaluate how different traits contribute to observations and how they interact. Finally, I'd say that genome-inferred traits with model integration does allow us to get at this, the search for generalizable rules. However, we're not there yet, and we need to approach this in a different way. And this is happening now. What we're seeing now is this scripted generation of trait predictions. Here's a nice example of a recent paper in Nature Scientific Data. But doing this based on harmonized data, so databases that integrate multiple sources of data, like NMDC is a good example of a group that's trying to make this happen. as well as facilitating open access to data workflows and models. And I think you'll see that K-Base is really pushing in that direction. So with that, thanks for hanging in there. And there's a lot of people to thank for this, and we're happy to take any questions.
Thanks, Owen and team. That was great. Excellent way to end the day. And we've had a ton of information. We did get some good questions in the chat. There was a lot of discussion over in Discord as well. But one of the questions that came from the Zoom chat was from Maria. She asked how can she predict the Boolean rules for a complex network? And is there any kind of program or software for that?
No, they essentially have to be coded in, like seeded from the literature. And I mean, the point with the Boolean rules is that it's flexible and simple enough to reconfigure, but they, as you heard this morning with DRAM, they are seeded from the literature by expert in that field. So the cases is an example of that. So where they map the presence of particular domains to substrates for complex carbohydrate breakdown. So those will be our rules. Yeah.
And then, yeah, there was a nice example from DRAM this morning as well, showing that the ability to use a substrate required both an exocellulase and an endocellulase, I think it was. So it's really, that is the synthesis of our biological knowledge creates those rules. What do we know about a process? And then we implement those as a rule. So we would provide a default, our view of what those rules are, but they're user adjustable decisions.
Okay. There was quite a bit of discussion over in Discord about inferring bacterial traits, especially genomes growth rate from genome information. And one question from Hitesh was how codon usage bias can be used to infer traits such as growth rate.
Well, maybe I'll have a go at that, and then Ulas can jump in. So codon usage bias occurs through the unequal usage of tRNAs. So take as a fast-growing organism will sample from the pool of available tRNAs in a biased manner. So more abundant tRNAs will be sampled. So that difference between the codons used versus the codons that could be used is directly related to the rate of growth of the organism. Now that's imprinted over evolutionary time. There are other factors that contribute to differences in codon usage, but it's been shown in a number of studies to be a good proxy for the upper bound on growth, so maximum specific growth rate, not realized growth, which varies widely, but the upper bound on growth rate. Other studies have shown that ribosomal RNA operon copy number is also a good proxy. But from work done previously, the codon usage bias seems to have more explanatory power. But as always, with more genomes and more phenotype data coming in, those associations need to be recomputed over time.
Okay, great. One last question from the Zoom chat. Young Chin asked, can you please explain more about the need of understanding traits at the species level versus in a pool manner from the angle of better understanding biogeochemistry of the system?
Yeah, that's a good question. Maybe I can start and then I'll hand that over to Gianna. So we begin with the individual. We want to know how the individuals work. But then if you zoom out from the individual and you look at a population, what you want to know is what are the generalizable features of organisms that are fit under these conditions, and what are their roles in biogeochemical cycles. So as long as you can make that simplification, that step of simplification, then you can scale from individuals to populations, and then you can do the same thing to scale to communities. What we'd like to be able to do is identify what are the generalizable properties of organisms that inhabit certain environments, for example, just like we do with plants. We have plants that are defined based on their growth strategies, whether they're opportunistic or conservative growth strategies, and that's directly related to observable traits like leaf area, for example, things like that. We want to be able to identify those same generalizable traits in microbes and then see how factors and properties of the landscape, like predicted inputs through vegetation, through photosynthesis, etc. Do those traits that we see in vegetation, are they also mirrored in similar types of microbial lifestyles? So by making those traits more generalizable, we can start to ask those questions beyond the individual to the community. And Gianna, do you want to add to that?
I think the information that we have is essentially from genomes is at the scale of single organisms. So at the scale of the single organisms, given biophysical constraints, we should see trade-offs between traits. And then also some biophysical models, for example, for the uptake of substrate properties in diffusion limited environments, they also consider like emergent affinities on the scale of single microbes. So yeah, in terms of model parameterization, there's a greater opportunity on more data to look at the single scale level and then see how this scales to populations or communities.
Okay, well, I think that kind of wraps up the questions from Zoom. There's an ongoing discussion right now over in Discord, and definitely encourage people to continue that as long as you'd like. And that channel, of course, will be open 24/7. Also wanted to mention that, well, of course, the presenters and the instructors and everyone has put in a huge amount of work, and I really want to thank everybody and appreciate the efforts. It's been a fantastic day from my point of view, and I would like to encourage, if any of the observers or students or instructors for that matter have any feedback for us. I made a feedback channel just now over on Discord, and would encourage you to put your comments, suggestions, questions, kudos, anything else in there. And really look forward to seeing you all again in the morning. So what we'll be doing tomorrow is we'll be moving from the metagenomics topic to metabolomics, and talking a lot about organic matter chemistry in the environment and how that's characterized and how that influences microbial community function. So unless any of the other organizers have any questions or comments at this point, it's time for us to adjourn. I appreciate we've done a pretty good job on staying on track today with time, and thanks again to all the speakers and instructors.